Hello friends, welcome to Medicines of 101 and today we will go through the topic that is very from the aspect of the biochemistry, that is the amino acid metabolism. So let's start the topic here today. Amino acid metabolism. Amino acid metabolism. Now guys, to understand the topic, last I'm just assuming there's, for suppose there are muscular cells, and I'm just going through the muscular cells for examples. These things happens in tissues, but I'm just taking the example for muscular cells. So let's assume it's a muscular cell. I'm just taking, for suppose it's a whole muscular cell here. I'm taking this as a muscular cell, the whole the things here. Just assume this whole as a muscular cells, and within the muscular cells what will happen, we have to know here that within the muscular cells there we are having what, one type of the amino acids that is called alanine. That's called alanine. Within the alanine we know it only what, that CCN, or we can say C. Okay, there will be one type of the amine group here, NH2 positive here, and if we say there's the CH3, the methyl group, there will be one more hydrogen, and there is acid group C. That's our alanine. That's our alanine. Okay, guys. And next, this alanine will get combined with another type of the keto acid that is called alpha ketoglutarate. That's alpha ketoglutarate. Okay, guys. What about the alpha ketoglutarate? Okay. So C double bond O, C double bond O, O negative, that means acid group is a keto group, that's why alpha keto. Then we are having what, CH2, and we are having a what, C double O minus. Okay guys, now we got this type of the combinations. Now there is enzyme that will work on them. What's the name of the enzyme? You have to know the name of the enzymes here. It's very important to know the name of enzymes. Now this it's an enzyme. Now this enzyme is having three pockets. There's the on pocket, there's the second pocket, and there's the third pocket. Now what? Within the first pocket, it will take the alanine. Just take it as alanine. And within the third pocket, first of all, it is taking the alpha ketoglutarate, because you know it's only that is the alpha ketoglutarate. Alpha ketoglutarate. Whereas, and what about it? It will take the alpha keto in another pocket, and there's within it there's like the ship based reagent. You can say, what's the name of this? Like the pyridoxal phosphate. Pyridoxal phosphate. And this is like the vitamin B6, along with it like the coenzyme cofactors along with it. Anyway, now these enzymes, the name of the enzyme is like, it's very important. You have to know the name of its ALT. What's the name of the enzyme? ALT. Or we can say what? We can say alanine amino transaminase. Alanine amino transaminase. Okay guys, now this enzyme is having such capacity, it will take the keto group from the alpha ketoglutarate, and it will just bring the keto group where, within the amine group, and it will take the amine group from the alanine, and it will just take the amine group, and it will bring where, it will bring within the keto group. Where was it, the keto group? There will like exchange in between the amine group and the keto group of alpha ketoglutarate and the alanine. And after the combinations, we will get two type of the compounds that we have to know here, and all the processes that is carried out by a single enzyme that is called ALT, or you can say alanine amino transaminase here. Okay guys, now let's see what can we get here. Now whenever this this keto group will come here, we will get two type of the compound. First compound, there is a C here. Okay, C, and there will be a keto group here, this keto group for suppose, and there is like what, CH3 here, and we had C double bond O, O negative. And is the structure like what? Pyruvate. Is the structure like pyruvate, or you can say pyruvic acid. Okay, so we can see what is it. So we are getting what by the combination of it? Pyruvate. Okay, and another thing we will get what? Plus, what did I say? There's this amine group. We will go where? In this position. This amine group will go here. Okay. Now, if this amine groups goes here, then what can you get here? See it here properly. See, no, NH2. This NH2 will come here. NH2. Okay. Then we'll get what? After that C double bond, whatever it is, just write it here. O minus here. Then we are getting what? It is CH2, CH2, and what? C double O minus. Now guys, is the structure for whom? Glutamate. Is the structure of glutamate. Glutamate, guys. It's another type of the amino acid, know it only. Now after the, I'm just repeating again. At first I'm just taking it's a muscular cells, and within the muscular cell the alanine on type of the amino acid, it will get combined with alpha ketoglutarate, and by the action of the enzyme there is like ALT, or you can say alanine aminotransaminase. Okay, and in this enzyme is having three such pockets. That's for alanine, one for alanine, another for pyridoxal phosphate B6, vitamin B6 cofactor here, and the another pocket for alpha ketoglutarate. Now this enzyme is having such capacity, will take the amine group from the alanine and it will get combined with alpha ketoglutarate, and it will take the keto group from the alpha ketoglutarate and it will get combined with whom? With the alanine, from where the amine group was came here. Okay. Now after the combination we will get two type of the compound, that is one is pyruvate and another is glutamate. One is pyruvate and another is glutamate. Now what will happen, guys? First suppose, okay, is the muscular cells. Muscular cells. Now what can happen for the pyruvate? The fat of the pyruvate may be what? The fat of the pyruvate may be here. It may be converted into there is lactic acid. The lactic acid, or it may be converted into the acetyl CoA. Acetyl CoA. Now this acetyl go through the TCA cycle. Then it will go to the electron transport system or chain, then it will produce ATP here. It will produce ATP. Okay guys. Now another thing for the pyruvate, it can also get converted into the lactic acid. Then what will happen? First of all, just assume there's like the blood vessels. It's like the blood vessels, and this lactic acid will be taken within the blood vessels. First of all, in the blood vessels, this lactic acid will come where? In the within the blood vessels. And first suppose I am just taking one more peak. You have to know here that first suppose the liver. I am just taking this as like the liver here, or hepatic cells. Okay guys, so first suppose it's like the, I'm just taking it as the hepatocytes or liver cells. Now what will happen? See it properly. Now this lactic acid, that is within the what? Within the lactic acid within the blood vessels, so that will go to the where? It will go to within the liver. First of within the, it will come within the liver cells. I'm just writing it in liver. First of liver or hepatocyte cells. Okay. Now what will happen? We got the lactic acids and the lactic acid within the liver. It will get converted into what? Again pyruvate. Pyruvate. By the enzyme lactic acid dehydrogenase. By the enzyme lactic acid dehydrogenase. So what? We are getting the product as the combinations of the alanine and the alpha ketoglutarate, the pyruvate, and the pyruvate will be taken within the blood vessels, and from the blood vessels this lactic acid will come into the liver, and within the liver by the action of the enzyme there is the lactic acid dehydrogenase. It will get converted into what? The pyruvate. What did you see here? Pyruvate, same thing here. Now guys, what? This pyruvate, whenever this convert, it will just what? The NAD positive will get converted into NADH. NADH. Okay, then the pyruvate will be here. Then this pyruvate can go to the what? Into the it can go to the there's enzymes like you can say PFCK. It will go to the glucose 6 phosphate. Up to like very many steps are there. So okay, first of all it will go to the glucose 6 phosphate. After that there is one more enzyme. There is like glucose 6 phosphatase. Glucose 6 phosphatase, by which it will get converted into what? Glucose. It will get converted into the glucose, and this is the process by which we are getting the glucose from the non-carbohydrate sources. We are getting the glucose from the non-carbohydrate sources. So we can say what? This is the neoglucogenesis. This is the process neoglucogenesis, in which we are getting the glucose from the non-carbohydrate from the amino acid sources. Okay, guys. Now we got here what? The pyruvate, and we got also the glutamate. Now what about the glutamate? What will happen in case of the glutamate? Now we have to know here. Okay. Now within the liver cells we got what? The glutamate here. Okay. See.